All right, guys. So we're gonna go ahead and get started. Hold on. Of course, I didn't grab my packet. Hold on, 1 second. Okay, so first thing I want to do, I don't remember if I reviewed osmosis and transport mechs with your class, some chapter 3 stuff. So I want to go through and do that really quickly because we have the time to do it today. And I realized a bunch of my students were telling me that the chapter 3 recording was a little confusing towards the end. So it should only take I don't know, 15-20 minutes max. So let's just go through and do that to start. I think that 60 should be close. Okay, so talking about movement through the cell membrane. All right. You have two types of transport mechanisms that we are going to talk about. Now, for the different types of transport mechanism, okay, for passive transport, right, there's 4 different mechanisms. We are going to focus on one. We briefly talk about the other ones, so that's why I've typed them out in your packet, but you don't need to know those as much as you do osmosis for the purposes of nursing. When we come across them later on in the semester, for example, we're going to talk about filtration when we get to the kidney, we'll get to them. You know what I mean? But for right now, just know whether they're passive or active, if that makes sense. I'm not going to ask you to identify the exact mechanics of, Mike, if it'll work today. Good. I'm not going to ask you to identify the exact mechanics of diffusion, but I might ask you something like, is diffusion active or passive? If that makes sense. So I want you guys to be identifying if they're active or passive, but beyond that, I really want you to focus on understanding osmosis. All right. So passive transport. When you are passive, you are not using energy. All right. So passive transport requires no energy. So for that little table thing here, this is what we're filling out right now, chapter or page 5. Okay. Like I said, I don't remember how in depth I went with you guys. I know that because I didn't record on Monday. I know that some of my classes I stopped at cell division, and then I think with you guys I might not have even really gone over this too much because you guys are my class that I had online before I got too sick. So passive transport, no energy. When you are being active, right, you are using up energy. So it makes sense that active transport requires energy. Okay. Passive transport does not require energy. Passive transport is going to have 4 different types. You have diffusion, osmosis, filtration, and facilitated diffusion. 4 types. Active transport, you only have 2 types. You have pumps and vesicles. Okay. So type it in chat. Actually, I think I could just copy this, put it in chat for you guys. Yeah. So like I noted on the first slide that I had left up, I will be checking. You might need to log out and log back in, Kimura. Destiny, please click on the screen. Otherwise, it's not going to count towards your MCQs. I'm going to be looking at the MCQ percentages. That's fine. Today. So make sure that you are clicking into the screen. It's week 3. So by now everybody should be at least a little familiar with Engagely, which is why I've kind of just been given some leeway with checking that percentage, but we really need to get to the point where we're doing that. All right. So type of transport that requires energy, active transport, right? If you are being passive, you are not using up energy. So type of transport that requires energy, active. Now, starting with two different types of, you might need to leave, oh, right. You can't hear what I'm saying. So diffusion is going to be like if you drop a dye tablet, okay? So, or like an Alka-Seltzer tablet or something, right? It's going to dissolve and then it's going to spread out because you want the concentration to be consistent throughout whatever the solution is that you're looking at, okay? So the goal is to move from that high concentration, so that like packed dye tablet, to a lower concentration so that it's like spreading out, right? Because like in the areas that the dye tablet hasn't spread to yet, it's a low concentration of dye. Osmosis, on the other hand, this is going to happen when those solutes cannot cross a membrane. Okay, so it's drawn to one side or the other. So it's going to move from a low concentration to a high concentration because those solutes can't spread out. All right, so that is why we are talking about osmosis, is the movement of water. Okay. So osmosis involves the movement of water from an area of low to an area of high concentration. Okay, so for those fill in the blanks, I typed them in the chat for you. Okay. Involves the movement of water, very specifically water. Okay, because water is the only thing that can cross that membrane. The goal is the same thing as with diffusion. You want concentrations to be on both sides, right? But chapter 3, page 5. This is for the notes packet. The study guide is very similar, like overlap wise, but I don't do the study guide in class. And I'll explain that more when we start like doing the multiple choice reviews. Okay. So if you think about it, chapter 3. So if you think about it another way, right? There's two ways to make something more concentrated. Okay. You can either add more solution to it, or you can boil off water, right? So when you are pushing things to one side, you are getting rid of some of that water to make the concentrations equal. Okay. So osmolarity is looking at the total number of dissolved particles in solution. All right. Now, tonicity, on the other hand, we are talking specifically about how that solution is going to affect the movement of water across a cell membrane. Okay. So tonicity, how a solution affects the movement of water across a cell membrane. This is only going to depend on solutes that can't cross the membrane. So non-penetrating solutes, because otherwise the solutes themselves would move across the membrane, right? In this case, we are looking only at the solutes that cannot cross the membrane. So for that little circle. Now that movement of water across the cell membrane is going to cause the cell to like change shape sometimes, or it stays the same. Okay. So this is especially important when we talk about nursing in the context of like IVs. All right. So when something is isotonic, does anybody know what iso means? Medical terminology. Annette?
Isotonic. Does it mean something like high?
So hyper means high. Iso means same.
Is that like the same?
Yep, exactly. So iso means the same. Okay. So in the case of isotonic solutions, all right, the concentration is going to be the same on the outside as it is on the inside. So, of course, my close button is like right over where I'm trying to highlight. So isotonic solution is going to be the same on the outside as it is in the cell. All right. Since it is isotonic, meaning that it is the same on both sides, the water is going to move into and out of the cell at an equal rate. All right. Since it is moving in and out at an equal rate, the cells remain normal in size. So they don't change size. Okay. So for your like fill in the blanks, number one is going to be same. Number two page is going to be equally into and out of the cell. Number three, cell stays the same size. Okay, so knowing that iso means same, it's going to be same across the board. Okay. For hypertonic, as Annette just said, right? Hyper is going to mean high. Okay. So hypertonic solutions have a higher concentration of solutes on the outside of the cell. All right. Osmosis, the water is moving from low to high, right? So it is moving from inside of the cell where it is lower to outside of the cell where it is higher. So water will diffuse out of the cell. All right. When you are losing water, those cells are getting shriveled up. So shrinking. Now, when I started this, right, I said there's two ways to make concentration higher, right? You can either add more solute or you can get rid of water, right? So think about like when you're like really dehydrated and you pee, right? And your pee is really, really concentrated because you have low water. So do you think hypertonic solutions are happening when you're overhydrated or dehydrated?
Question you asked, could you repeat that?
So hypertonic solutions, right? We said that you're losing water. Well, so there's two ways you can increase the concentration of the solution, right? You can either add more salt or add more whatever it might be, or you can boil off the water. You can decrease the water content. So hypertonic solutions, you have a higher concentration of solutes, right? So do you think it's happening when you're overhydrated or dehydrated?
Dehydrated.
Dehydrated, right? You are losing that water. Dehydration. Okay. So it happens during dehydration. Hypotonic solutions. Hypo means low, right? So like if you're hypoglycemic, you have low blood sugar. Oh, let me type this out. For hypertonic, one is going to be higher concentration, two moves out of the cell, and then three shrinks. Okay. In hypotonic solutions, all right, it is going to have a lower concentration of solutes. Therefore, the movement of the water is going to be out of the cell. Or sorry, into the cell. I was like, wait a minute, that's wrong. Yes, right? Because we are moving from low concentration to high concentration. So if it's higher inside of the cell and lower outside of the cell, it's going to move into the cell. This is going to cause the cell to swell and eventually burst. Now, if hypertonic solutions happen in dehydration, hypotonics are going to happen when?
You're hydrated.
Exactly, overhydration. happens in overhydration. Okay. Now, this is why we don't put water, like pure water, into IV bags, right? We put saline solution in IV bags. Okay. So this is why. If you do not have any solutes in the water, right, it's going to move into your red blood cells and cause those red blood cells to burst. This is why osmolarity is really important when it comes to nursing. So and then 3 swells. Okay.
Hey, Brittany,
Mm-hmm.
could you give me to the number 1 and 2
Like for isotonic?
diffusion
Oh, for diffusion, there's nothing to write down.
for the, osmosis is just the water. It's a movement of water from an area blank to area blank.
Yeah, so it's an area of low concentration to an area of high concentration. I typed it out in chat, but it looks like there were a couple of
It's an
chats
area,
afterwards.
to an area of low to an area of high.
Mhm. Yep,
Okay, thank you.
no worries.
Mm.
All right, so that is osmosis. It is important when you talk about IV bags. So when they talk about different concentrations of saline solution, what they're talking about is different concentrations of salt in that IV bag. Another graphic to help you guys. This video is really good. I believe I played it for you guys to like show you different examples of osmosis. Now, next you have filtration. The easiest way to explain filtration, there's nothing to fill out for it. Okay, the easiest way to explain filtration is a coffee pot. Okay, you have your coffee pot inside the filter, right? Water is going to push through the coffee pot. As it pushes through the coffee pot, pieces of the coffee are going to dissolve, but the ones that are too big get held back, right? So the ones that are dissolved in the water going to get pushed through. The ones that are too big get held back. We will talk about filtration when we get to the urinary system towards the end of the semester. For now, I just want you guys to know that it's passive transport. While you guys answer this, I'm just going to grab more water. Should take me about 30 seconds. Okay, so let's do about 10 more seconds. Exactly. So filtration is going to be a type of passive transport. Okay. So it does not use energy. The last type of passive transport we have is facilitated diffusion. It is just like diffusion.
I have a question.
Mm-hmm.
Where is everybody answering the questions at? Like, I'm confused.
So they just show up on the screen and you just click on the letter.
Oh, okay. Okay. Thank you.
Yeah, no worries. So facilitated diffusion is just like diffusion. The only difference is that sometimes you need like a little carrier protein, meaning that you just need like some help. Okay. A carrier protein is just there to help it out. To facilitate, right? So next we're going to talk about active transport. Okay. Active transport mechanisms. I left the fill in the blanks just to like, I don't know, help focus. But again, I want you guys to be identifying whether it's active or not. Okay. What active means, meaning that it uses energy, right? When we get to these mechanisms, we will talk about them. So in active transport, solutes are going to move up the concentration gradient. So meaning from areas of lesser to greater. So you can think of it as like swimming upstream, right? It's harder to swim upstream. So it makes sense that you are using more energy to swim upstream. So active transport is going to require energy in the form of ATP. Anytime you hear ATP, you should be thinking energy. Okay, so I'll type this all out in chat for you guys.
We fill in the blanks.
Yep, I'm typing it out.
Oh.
So we have two types of active transport mechanism. Okay. So you are going to have transport by pumps. The most important example is going to be the sodium-potassium pump. Transport by pumps. What's it? Okay. So this is going to move sodium out of the cell and potassium in. And uses ATP to make the exchange. All right. Now, I do not expect you guys to know a lot about the sodium potassium pump yet. All right. It's going to come up when we get to neurology, neuroscience. I forget what it's called. Nervous system for you guys. Okay. When we get to the nervous system, we will talk about the sodium potassium pump in more depth. Well, not in more depth, honestly. This is all you guys will need to know for it, really. Okay. The sodium is moving out, potassium is moving in, uses ATP. Okay. Do not worry about knowing that it moves 3 sodiums out, 2 potassiums in, and okay. Just know for now that pumps are active transport mechanisms. Next, you have transport by vesicles. We've talked about vesicles before, right? So vesicles are just packages. Okay, so we talked about vesicles when we talked about the Golgi apparatus. They're just packages, that's it. So vesicles are going to transport matter into yeah, so I'll copy this and put it in chat. Highlighting everything. Transport by vesicles. Okay. Okay. So cells also have the ability to move large particles or numerous molecules at once through the plasma membrane. Uses vesicles. So anytime you hear vesicle, just think package. Okay. You have 2 types. Does anybody know what the prefix exo means?
Would it be Out or
Exactly.
exit?
Exo means exit. Okay, so if you think of like exoskeletons on bugs, right? Outer, or like exo means exit. Okay, so exocytosis releases substances out of the cell. You see this a lot in glands. For example, mammary glands. Okay, so mammary glands, they make milk, right? Milk does not diffuse through plasma membrane, meaning it doesn't just go through. You need some other way to get it through. It's too big to go through. So mammary glands are going to make this little package, and it is going to push it outside of the cell. Okay, so it's going to put the milk into a package, and it just like, what happens is like, you have your package here, and it merges with the plasma membrane. So if my arms are the plasma membrane, and then it just kind of shoves it in or out in this case, but I can't like do it this direction because I'm not double jointed. So, okay. So exocytosis releases substances out of the cell. Endocytosis, if exo means out, endo means in, right? That's it. So endocytosis is going to bring substances into the cell. Into the cell. Okay. So plasma membrane traps substances that are too big. Or too large to diffuse the plasma membrane and brings it into the cell. Okay. So those are your transport mechanisms. Again, when we get to exo and endocytosis, we'll talk more about them. For now, I just want you to know transport by vesicles is an active transport mechanism. Okay. I also want you to know that vesicles means package. You'll start getting more familiar with exo means out, endo means in when we get to like more medical stuff. But Annette?
So basically for transport, expo, and sodium, all those is active,
Yep,
active transfer. So all those is under active transfer.
exactly.
Okay.
All right. Okay, 10 more seconds. Yeah, exactly. Sodium potassium pump is going to be active transport, okay, meaning that it uses energy. All right, 10 more seconds. Yeah, exactly. Osmosis involves the movement of water. Okay, so you're moving water across a membrane. Next, we're going to talk about cell growth and reproduction. So there's a lot of slides in here with a lot of text. I like to focus on this table because I think it summarizes everything pretty nicely. So I'm going to type it out in chat for you guys so that if your text is a little bit small from the table, it is on here. Okay. This was your final Jeopardy. Oh, wait, you guys didn't end up playing Jeopardy. We're going to play it today if we have time. I think I figured out a way to like play it remotely. We'll see. Okay. So DNA, the function of DNA is going to be to store genetic information. Okay. So I always use the textbook in the library analogy, right? So you go to the library, you want to build a bridge or something like that. I don't know. So you need to get a manual on how to do it. All right. In order to do that, you're going to find this ginormous textbook, right? But the textbook is too big to check out of the library. So what you then do is you're going to make copies of those pages that you need, right? So, copies and transmits. Is there a way to, yeah. RNA, copies, and transmits. Okay. So RNA is going to be the copies. So you only need like one chapter from the textbook, right? So you make copies of the pages that you need. That is going to be your RNA. DNA has this structure that's like a twisted helix thing, okay? In DNA, you have 2 strands. RNA, you are only going to have 1 strand. I just realized my door's open. Otherwise, like those. So when we say double versus single helix, what we're talking about is the number of strands. Okay. So DNA has 2 strands, RNA has 1. So D with D, R with single. I don't know. Composition. You do not need to memorize deoxyribose. Deoxyribose is a long word, right? So all you need to know, it's a D sugar. All right. If it looks like a long, complicated word and it starts with a D, probably goes with DNA. All right. D stands for deoxyribo, Na stands for nucleic acid. That is the same between DNA and RNA. Okay. So if you're looking at a question for what does DNA stand for, or RNA stand for, D sugar, nucleic acid. RNA, on the other hand, has an R sugar. So ribonucleic acid. So R sugar. For the bases, you'll notice like the first 3 are the same, right? So when you're studying for a test, you don't want to focus on what's the same, because chances are when somebody asks you a question, right, you want to go for whatever like the keyword is. In this case, what is different between the 2? All right. So I would listed out the first 3 for you, but knowing that DNA has thymine and RNA has uracil is going to help you, right? Because if we ask you, where is uracil found, right? We're not going to ask you where cytosine is found, because cytosine is found in both RNA and DNA. And lastly, just know that the DNA is found in the nucleus. Do not worry about knowing that it's found in mitochondria. Okay, that's a whole nother thing. So DNA is going to be found in your nucleus. RNA, so the nucleus in the analogy is going to be your library, right? So DNA is found in your nucleus. RNA is going to be found in your cytoplasm. Okay. So if you're having a hard time reading the table because it's like small on whatever, I typed it all out in chat for you. So you can use that.
Quick question.
Mm-hmm.
Oh, before you do the test, can you go back?
Sorry, hold on. It acts, so I can't, like, if I stop it, it's going to count against everybody. But it's typed out in chat. What was the question in it?
It was for sugar. So sugar is the deoxynic acid, and the other one is the
Ribose. Yeah. So if you look at chat, it's typed out too. It says sugars, DNA is deoxyribose, RNA is ribose.
and the last base. Oh, okay, I see it. DNA. I see it. Thank you.
Yeah, no worries. Okay, let's do like 10 more seconds. Yeah, so DNA has a double helix. Okay, so double meaning it has those two strands. I don't know if you guys got it right, all because I accidentally flashed the question or the answer beforehand, but I'm gonna give you all the benefit of that. D goes with double. Okay. Protein synthesis. The only thing I want you guys to know about protein synthesis, there are two phases. They are transcription and translation. Ricky, it's typed out in chat. So, protein synthesis. I can type synthesis. So, making proteins, transcription, and translation. Okay. If I ask you, is mitosis part of protein synthesis? You guys are going to say no. If I ask you, what are the two phases of protein synthesis? You guys are going to say transcription and translation. Okay? That's it. I do not expect you guys that your book has this whole big diagram. You guys do not need to know anything from this diagram other than protein synthesis includes transcription and translation. That's it. Okay? Do not kill yourselves trying to memorize that diagram. If you're like curious, you can go in and look at it. It's there for you, right? Do not worry about knowing anything other than protein synthesis is transcription and translation. Okay, our last thing. Oh, wait, hold on. Do another MCQ.
What do you put for the 1 and 2 box on page 8?
Transcription and translation.
Transcription first one, translation second. Okay.
Yep, that's it. Let's do like 10 more seconds. All right, so uracil is found in RNA. Great job, guys. So uracil is going to be found in RNA. Okay. Jasmine, just click on the screen.
And Miss Brittany, I'm sorry, girl, I was late because I had to get somebody to keep the twins real quick so I can hop online. I had to drop them off at my auntie house.
Oh, okay.
Thank you. I'm sorry about that. And I had got the message from the dude on Pan Open, but that stuff still ain't working. So I'm waiting on him to contact me back. It let me in, and then when we was talking about how to set it up, he like, go back and log in when you change the passcode. And then it kicked me right back out. It keeps saying that my instructor got to send me a shared link.
Okay. Shoot me an email, or shoot me and Dr. A an email. So I think we CC'd you on a thing, because she'll probably know more about what's going on, because she's the one that like just talked to them about all the error messages that came up.
Okay, you said that's shoot you a message, and who else?
Dr. A, I think she was the one that talked to you before about everything. As
No,
well?
a man.
Okay.
Yeah, man. I never know Dr. A, just you and the IT guy.
Oh, okay. Shoot me an email.
Okay, cool.
I'll
Thanks.
forward it along to her.
Okay.
Carmen, did you have a question?
No, my bad. That was an accident.
No worries. I just try to check in if like somebody raises their hand. So. All right. So transcription and translation are two phases of protein synthesis. Okay. So 2 phases of protein synthesis, transcription and translation. That's all you need to know. Okay, so next we're going to talk about the cell cycle. The cell cycle has a whole bunch of different phases. Do not worry about knowing these. The only one that I want you guys to know is what mitosis is. Okay? So in mitosis, we are talking about cell division. Everything else is just like the cell existing. Okay? So mitosis is just when the cell splits into 2 identical daughter cells. All right. There are going to be 4 phases of mitosis. The first one is going to be prophase. I don't expect you guys to know too much about like the phases. Okay. If you've ever heard of like chromosomes, so like I'm sure a lot of people have heard of Down syndrome has like the extra chromosome, right? Prophase is when the chromosomes are made. That's, I mean, you don't even really need to know that. Just know that prophase is the first one. Okay. Metaphase is going to be the second one. During metaphase, the chromosomes line up in the middle. So I always say metaphase middle. Anaphase, the pairs of chromosomes. So right, we're making 2 cells, which means we're copying everything. So the copies of those chromosomes pull away from one another. And then lastly, you have telophase. Now, which is when the cells finish separating. Okay. Now, I am fully aware that if you guys Google mitosis for some reason or another, there are technically 5 phases, as many as 7 I've seen listed. You guys just know mitosis needs 4 phases in this class. There are 4 phases of mitosis for the purposes of this class. Okay? Exactly. KeyMat. That's it. Okay?